Hello everyone, welcome to my class. Today we will talk about nucleic acids. So what are nucleic acids? Nucleic acids are simply the biologically important polymers which are present in all living beings, and they are really essential for the genetic information. They help in transferring the genetic information from parents to offsprings, and they control our heredity. So basically they are involved in information storage, in protein synthesis, and in energy transfer mechanism. Now next is chemical composition of nucleic acids. Actually nucleic acids are made up of three main parts, three main units. The first unit is a nitrogenous base, the second unit is pentose sugar, and the third unit is phosphoric acid. Let's talk about nitrogenous base. Now there are two types of nitrogenous bases which are present in the nucleic acids. Number one is the purines and number two is pyrimidine. Purines and pyrimidines are two nitrogenous bases. Now purines are adenine and guanine, whereas pyrimidines are cytosine, thymine and uracil. There is a basic difference between purines and pyrimidines. All purines are bicyclo compounds, whereas pyrimidines are monocyclic, but both are heterocyclic molecules. Heterocyclic means other than carbon, there will be nitrogen or oxygen also. Now coming to the sugars, there are two types of sugars which may be present in the nucleic acids. Number one is 2-deoxyribose sugar, also called beta-D-2-deoxyribose sugar, and another molecule is ribose sugar or simply beta-D-ribose sugar. The common thing is both of them contain a 5-membered ring, and it's a heterocyclic ring, but they differ with respect to the second carbon. In case of 2-deoxyribose sugar, oxygen is not attached with the hydrogen at carbon atom number 2. Whereas in ribose sugar at carbon atom number 2, there is OH group. So on the basis of the sugar, we can have DNA, that is deoxyribose sugar, and RNA, that is ribose sugar. Means RNA contains ribose sugar. Of course there will be purines and pyrimidines, but in DNA there will be beta D2-deoxyribose sugar, plus there will be nitrogenous base, and there will be the phosphate group, phosphate. Now what is nucleoside? Nucleoside is base plus sugar, means when sugar is attached with the base means nitrogenous base, then the resultant is called nucleoside. So it's a combination of base and sugar. But what is nucleotide? Nucleotide is, it's a combination of base, sugar and phosphate. So that means the basic difference between nucleoside and nucleotide is the presence of phosphate group. When phosphate get attached with the nucleoside, then it is called a nucleotide. So nucleotide is a combination of sugar, nitrogenous base and phosphate group. Now next is, how are different nucleotides attached with each other? Now different nucleotides are attached with each other through a phosphodiester linkage. A phosphodiester linkage or bond, it occurs when exactly two of the hydroxyl groups in phosphoric acid react with hydroxyl groups on other molecules to form two ester bonds. That means here we need to connect two nucleotides via this bond. And how are these nucleotides will be connected? They will be connected via phosphodiester linkage, which will be obtained by the loss of water molecule, and it will be forming two ester type linkages. In this diagram, there is the formation of phosphodiester bond. So coming to now the structure of DNA. Now DNA has got primary as well as secondary structure. Now in the primary structure of DNA, in the DNA, all the nucleotides, they are connected via phosphodiester linkage. So DNA is a macromolecule and it consists of two polydeoxyribonucleotide chains. And each chain is formed by linkage of many deoxyribonucleotides through phosphodiester linkage. Coming to now the secondary structure of DNA. Actually, the structure of DNA was proposed by Watson and Crick in 1953, and they proposed a double helical structure for DNA. According to them, DNA molecule consists of two deoxyribonucleotide chains that are spirally coiled around each other on a common axis. And this coiling is generally a right-handed coiling. The diameter of DNA molecules is uniform throughout. The backbone of this double helical structure is made up of phosphate and sugar. Means it's a phosphate sugar moiety. The double-handed helical structure, it is right-handed helical structure. Antiparallel strands, they run 5' to 3', and the backbone is made up of sugar and phosphate. In this bases are present and bases they point inward. Bases of opposite strands are combined together through hydrogen bonds. Between cytosine and guanine, there will be a triple bond. Between adenine and thymine, there will be double bonds. Now there are major and minor groups also. And in DNA, complementary base pairing rule is followed. In DNA, bases are 0.34 nanometer apart in a strand and 3.4 nanometer or 34 angstrom per turn of the helix. In one complete helix, there are 10 nucleotides present. So that means helix is having, in helix, the two nucleotides, they are at a distance of 0.34 nanometer. And the total diameter of the helix is just 2 nanometer or 20 angstroms. So coming to what is complementary base pairing rule. Complementary base pairing rule means that one purine can only combine with one pyrimidine. No two purines, neither two purines nor two pyrimidines can be accommodated collectively in the DNA helical structure because of the exact spacing. So adenine always pair up with thymine and cytosine always pair up with guanine. Adenine and thymine, they join through double bond, whereas cytosine and guanine, they join through a triple bond. So this rule is called complementary base pairing rule. Means if on one strand, let's say the sequence is A, T, A, T, A. Then on the other strand, the sequence will be T, A, T, A, T respectively. Means adenine will combine with thymine only and cytosine will combine with guanine only. Now before we start with RNA, what are the biological functions of nucleic acid? There are two important functions. Number one, it is involved in the replication process. So what is replication? The process by which a single DNA molecule produces two identical copies of itself. Replication is always catalyzed by a few enzymes, and it starts with the partial unwinding of the two strands of DNA through breaking of hydrogen bonds between the various pairs of bases. The second main function is synthesis of protein. Now DNA plays an important role in the synthesis of protein, and it occurs in two steps. Number one, transcription. Number two, translation. So what is transcription? The process which leads to synthesis of RNA is called transcription. That is, we are converting, we are using DNA for the synthesis of RNA. So this is called transcription. Now this transcription, it starts partially by the unwinding of the two strands of DNA double helix, again by the breaking of hydrogen bonds. Next is, what is translation? Translation actually starts in cytoplasm. So after transcription, messenger RNA moves from the nucleus of the cell to a ribosome in the cytoplasm, where it gets attached and serve as the template for the synthesis of protein. So that means in the translation process, the protein synthesis completes in cytoplasm. Now let's come to the three types of RNA. There are three types of RNA. Number 1, messenger. Number 2, transfer RNA. Number 3, ribosomal RNA. So messenger RNA, it helps in transcription. Number 2, ribosomal RNA, it helps in the protein synthesis. And transfer RNA is really essential carrier molecule for amino acids to be used in protein synthesis. Let's come to the difference between DNA and RNA. DNA is always double-stranded nucleic acid, whereas RNA is single-stranded. DNA, it contains deoxyribose sugar, whereas RNA contains ribose sugar. DNA contains thymine as a nitrogenous base, but in RNA, instead of thymine, we have uracil. DNA is the genetic and hereditary material of the cell, whereas RNA is involved in the synthesis of proteins. DNA is present in the nucleus of the cell, whereas RNA is present in both nucleus and cytoplasm. Coming to the last, that is DNA fingerprinting. No two individuals can have same DNA, identical DNA. But yes, DNA of parent resembles with offsprings to some extent. So that's why DNA fingerprinting can be used for the solving of dispute of parenting. Means parentage disputes can be easily resolved using DNA fingerprinting. And number 2, of course, it is quite helpful in solving criminal cases also. So it is being used in the forensic laboratories to establish the identities of individuals. And of course, for mutations also, we use DNA. For the formation of the hybrid crops and all that, we need DNA.